Thanks everyone to coming to my talk. Can you hear me clearly? Can you hear me?
Oh, yeah.
Okay. Yeah, usually people complain about my voice is too soft. Yeah. Today I will talk a little bit about my research in CAR-T immunotherapy. So first I will introduce about what is CAR-T immunotherapy. So basically, you might heard of this word CAR-T immunotherapy from some newses. It's kind of like, if you have $1 million, probably the cancer might be cured. But that's just like the advertisement for that. The CAR-T, it is a new therapy for the past 10 years as a groundbreaking therapy. You can see there are two plots about a summary paper talking about the great efficacy gain for the CAR-T patient, who, if you receive the CAR-T, their overall survival, complete response, and the PFS are all great improved. So those patients are not every patient having cancer. A specific population, especially the patient in blood cancer, they have multiple relapse. Their average, their median overall survival is just about 6 months. For those patients, the CAR is kind of like the last hope for those patients, and it works pretty well. So what is CAR-T? It is in general, it is a genetic modified technique that's modified patient's own T cells at a particular receptor on the top of the T cell, and use patient's own T cell infused back to patient's body to recognize the cancer cell and kill it. So basically here, you can see the yellow one is a T cell. It's like the policeman in our body, and the pink one is a tumor cell. So the hard thing is how to let the T cell to recognize the tumor cell and then kill the tumor cell. The CAR-T is in the lab, scientists add this particular called CAR chimeric antigen receptor on the T cell, and let the T cell can grow by themselves and infuse back the T cell to patient's body, and the T cell can recognize which one is tumor cell and kill that. Basically, give the policeman a radar, they can recognize who is the bad guy. So the workflow of the CAR is a patient come to a hospital, and then the doctor gets the patient's own blood and gets the T cell. Then they go to the lab to genetically modify the T cell, generate this one called a CAR T cell. The patient will also receive something called lymphodepletion chemo that is helping them to better receive the CAR. So this is one time point happened before the CAR-T infusion. So that's a pre-CAR-T infusion time point, very important in the CAR study. And then the patient will receive the genetically modified CAR cells back, get this infusion, and the CAR T cell will grow in patient body. So this is a plot about what is the genetically modified T cell looks like. It has sort of a receptor here, and the cancer cell, there is some different antigen. This particular CAR is called CD19. So basically, the CAR T cell recognizes CD19 antigen, but there are different versions of the CAR. It has CD20, CD22, or a mix of the CD19 and CD22. So the CAR T has a lot of different versions of the CAR T. Now we have 6 approved, FDA-approved CAR product. It's all treated for the blood-related cancer, but those products has more and more labels can treat multiple blood cancers. So this is the current growth. The more groundbreaking is scientists investigate some scientists investigate whether the CAR can work in solid tumor. It's harder to let the T cell recognize the cancer cell in a solid tumor and kill it, but more and more research is doing that, including the brain tumor. Some study I participate in has very strong response to the neuroblastoma, so that's a big improvement also for autoimmune. So some of the CAR therapy can treat for the lupus. So that's another groundbreaking news recently. So basically, this is the growth of the CAR. It is in the past 10 years, it's grown rapidly. So here is a summary about the number of the CAR trials globally. The two big countries, China and the US, has a lot of CAR study. You can see it's grow very fast. And you will see now China has even more trials than US. For the most of the trial, you will see the CAR study is phase 1 or phase 2 study. It's because of the FDA approved CAR trials happen at phase 2. So you don't need to design a phase 3 study to get approved. So there isn't going to that stage to have randomized the study to compare to product of CAR. Most of the CAR study is still single center phase 1-2 study. So the good thing is it's blooming a lot of trials, and the different trials can testing different combination of the CAR, different modification of the CAR structure. But the statistical issue, the challenging what I face is the sample size is very small, and the study is not a randomized study, it's single center phase 1-2 study. So I'm the lead biostatistician for more than 20 CAR-T study involved in dry Hatch and Seattle children. So at Hatch, the difference probably with other research institute is, as a biostatistician, I more take care for some of the disease area, and I'm doing the research from the beginning to the end. So I also do some statistical analysis for mouse model, preclinical work like RNA-seq study, chip sequencing. I'm mainly doing clinical trials as a lead biostatistician for CAR and some other disease. I also do the correlative study, secondary analysis. After we finish the trial, we analyze the data. So this is probably a main difference from Hatch and other institute. We work with I work with more clinicians than I work with my staff collaborator. So it's my honor to work with those world famous clinicians to help the CAR field. So this is one of the study I will talk mainly using the data today. It is a relatively large study, has about 200 patients, but has 3 different diseases. So we will do analysis separately. You can see for each disease subset, the sample size is about 50 something, and you will see the ALL, this patient subtype, they have a very high response. But for the NHL, the non-Hodgkin's lymphoma, the patient having different response rate for different disease subtypes. So one of the main questions the clinical challenging is everyone wanted to know why some patients receive the CAR, they have a durable complete response. They live longer, they don't have relapse. But some other people, they either has no response or they have initial response but quickly relapse. We wanted to know why. From how to do that is we do biomarker identification. So the CAR, we collect the blood frequently. We collect patient blood before the CAR-T infusion, and we almost collect every day after CAR-T infusion until the first 28 days. And we have frequently evaluation for the first 1 month, 2 months, 3 months, 6 months, 1 year. So we have more time point than the other study. The data challenging is dimensionality, because the CAR patient, as we see, we only have that much of the patient. I don't know how many patients being treated by CAR, but at least 3 years ago, the whole world, there are only 1,500 patients being treated by CAR. So the sample size is relatively small in the CAR field. But those patients, I kind of think that we frequently check them by different assays. So today we can use DNA to check about their DNA. And the next day we have a protocol to say, oh, let's check the single cell RNA-seq to get what's their marker. And we do carton run, we do cytokine at a different time. We do 2 different versions of the cytokine for the same time for the same patient. So we do a lot of things for those patients. The good thing is we have quite a lot of blood at the beginning, can do a lot of work, but on the other side, from the data analysis point of view, we have high dimensional data. So my summary is like, we need to use appropriate statistical method to analyze this type of data. So the part 2, I will talk about what I have done about the clinical collaboration. So the data we have is small sample size, about 50 subjects. And the biomarker data is high dimensional data. It has high dimensional covariate. It's incorporating the clinical characteristic, like what kind of treatment they received before, age, gender, and how bad about their disease. We also receive something called cytokine. That's basically all the CAR patients receive this cytokine assay at a different time point. It could be even before infusion, pre-lymphodepletion. That's one time point happened before they receiving CAR. And at the day 0, they receiving CAR, and the Cmax is the one of the time point when their CAR T cells in their body grows to the maximal. There is a sort of like a peak, a time usually happened after 2 weeks after they receive the CAR infusion. So at a different time point, we get the cytokine measures. Those data are highly correlated. I will show you, it looks like blocks in the correlation matrix. And also the signal is not sparse. So as a lot of people see in genetic study, you find the, one of the SNPs from the GIVA study, you try to find the needle in the sea. In the CAR study, it's basically you go to a garden, every flower is blooming. You just don't know which one is the most beautiful one. You have a lot of blooming flowers. So the CAR is like that. The signal is not sparse, a lot of significance. You don't know which one is the most important things we can control about their disease. Also, CAR has a lot of different endpoints, continuous binary time to event. I'm not investigating putting all the endpoints together, try to strengthen the signal to find the marker. I will analyze that separately, but my work is try to have a pipeline that can work for every type of the endpoint. So here is a correlation matrix about the X covariate. And you can see there is the red one is a cytokine, because the cytokine measure at a different time point. So this is a small block, is the cytokine measure at a different time point. So naturally it will looks like a block. And you can see it's not only within the cytokine, it has a block structure. Between the different cytokine, it also has correlation. Some correlation could be strong, some correlation could be negative and positive correlation either. So the cytokine structure is harder to analyze, and almost always when I find a cytokine, there is some biology explanation about why it's like related with why the disease happens. But so far, we tried so many different studies, and we didn't find a very robust cytokine or signature. We found some biomarker very strong predicting about what happened. But those kind of things not always happen exactly the same. So those another thing is how to try to find the robust signal. So the high dimensional data question what we have is we have 48 subjects and we have 132 covariate. How to do the high dimensional data analysis to identify those biomarkers. So the standard approach is stepwise regression. Like in this audience, probably everyone having a lot of to see, we shouldn't use stepwise regression. But in the clinical paper I reviewed, I would see 50% of the paper still use stepwise regression. In my talk, actually, I don't want to see stepwise regression is the end of the world. You know, you're using that, you are not losing everything. It is an approach. We can use it, but we need to know at what situation it performs good, at what situation it doesn't work very well. So we all know one of the major things about stepwise regression is you cannot directly use that to deal with high dimensional data, right? If P is greater than N, and it's well known to have this multicollinearity issues. So in one of the paper, the first paper we published, the biomarker identification, we use a modified stepwise regression. It is kind of like what everyone use about stepwise regression, but no one gave a name. I just call it a two-step stepwise regression. So the first step is we do the univariate analysis. And from the univariate analysis, we ranking the covariate with a p-value if it's less than 0.1, or, you know, like in my study, like it has a lot of significant covariate. So we have another threshold, we force the number of the variable into the next step is less than half of the sample size. And then we run the stepwise regression, use a covariate from the first step. So we have 45 subjects, and we tried this method. So here is the table one in my paper, like what we observed. So you can see here, we have a lot of covariate. If we use a threshold 0.1, it is significant. So then we only have 45 subjects. So you throw out a lot of covariate into one of the regression model. It is working, but it is not the best way to do it, right? So we use that two-step stepwise regression, and we throw that in and we selecting 3 covariate. That way, we eventually thinking those are clinical relevant covariate. So we also generating the Kaplan-Meier curve with those 3 markers. So the separation of the Kaplan-Meier curve, the threshold, a lot of my clinical collaborator is asking me how we should separate a continuous marker. And they always ask me whether I should separate that by median. I said, okay, median is, again, not the end of the world. Like you can do that by median for sure. But it's I would recommend mainly use your clinical knowledge to judge what will be the threshold. So here we, all the threshold we're selecting, it is sort of clinically, they already know that's relevant. So we separate that to be high risk and low risk, like the LDH platelet, and whether we use this additional lymphodepletion. So we got 3 subgroup from 3 biomarker, and we put all the low risk group together to be a joint low risk group, and we get the low risk and high risk group separatedly. Luckily, we have a good subgroup size, 33%. So then by this definition in our data, it looks reasonable. So this is one of the things we're suggesting for the other study also do is check out those 3 markers and look at the low risk and high risk. So the first one we do stepwise regression, 2-step stepwise regression. The second one we try to see whether we can use penalized regression. So Lasso and ElasticNet is a very popular method for our audience. I don't think I need to introduce too much about what is Lasso or ElasticNet. Just show like what we do about Lasso and ElasticNet modification. So the basic idea is why we're doing this. We call that frequency ElasticNet. Is the idea is very simple. It's like we believe you run Lasso, you run ElasticNet once, the result is not as robust as you run ElasticNet 100 times. So we are in one way doing the data analysis, we noticed that because of there is a cross-validation step to tune the parameter in ElasticNet. So when you're changing the random seed, your result might be dramatically different from the previous what you run. That's a big problem when you're doing the real data application, because you don't want every time you change a seed, you get a totally different result, right? So here we don't do any bootstrapping or subsampling. We just changing the random seed 100 times. And every time we use exactly the same data set and we do the ElasticNet again. So, and we run it 100 times and based on different random seed. And then we choose the final model based on a frequency table. Basically, if 85 out of 100 times we select those marker, we believe that should be better than we just run ElasticNet once. So that's the rationale. So we use this approach in one of the paper published on Blood. And again, this is sort of one way we give the modification, but whether this is the best solution, we don't know, right? There are some other high-dimensional data analysis methods. One thing I want to point is called stability from Peter Billman's group 10 years ago. And it is very similar idea as the frequency elicited too, but it has a very good statistical property. I'm not sure that everyone familiar with that, but it is in the package HDI. So it's very easy to use. The idea is they just do subsampling. Basically, they don't do bootstrap either, but they, if you have 100 patients, you can subsample 80% of the sample and build one model. You random subset 100 times with 100 different subsamples. Each subsample has the same sample size, 80% or 75%. Those are the parameters you can tune. And you run that 100 times and you apply the algorithm on the subsampling data set and you check about the empirical selection probability, basically the same. If you select some variable greater than a threshold 85%, those are the variables you finally will select. So we tried stability in our data set too. It has good statistical property, but it has also very similar issues as ELISA and LASSO. They need to refit the data. A lot of parameters need to tune, and we notice it's even more sensitive than LASSO or ELISA about that parameter because they have many parameters as an input parameter. So what's next? So mainly for this talk, I'm using the high dimensional inference method, what is published for 10 years now. So high dimensional inference, mainly I had two different versions, desparsified HDI and decorrelated HDI. So those two algorithms published for linear regression or general linear regression. So recently, not that recently, it has a time to event endpoint implementation for both of the methods. And also it has for competing risk model, it also has the implementation. So we're collecting all those algorithms and implement them into our Shining tool. I will show you quickly what it is. But the idea is of high dimensional inference. If you are not familiar with that, it is basically the first step you run a lasso and you get an initial lasso coefficient estimation. From there, you do the inference part. And that is, those two methods have different way how to do the inference. Eventually, it will give you for all the covariate, it give you a confidence interval and a bears the beta estimation and the p-value. So that can help you for the inference. Our research is to compare different methods and at the small sample size and highly correlated covariate, and we apply that for the CAR-T data. Okay, do you have any questions? So I would send to my collaborator to help me on this comparison. The simulation setup is we compare several regression, the frequency elicited 100 times elicited, the stability we also run 100 times, and the desparsified HDI, decorrelated HDI. We adopt the real data structure is we have the block, correlation matrix have a strong correlation as a block structure for those covariant. We adopt the sample size in our scenario. In our real data analysis, we have small sample size 50, 150, and we give a true biomarker to be 3 or 15, and the total number of the biomarker changing from 15 to 200 as to incorporating the high dimensional setting. We give access of the true beta basically different beta range and do 100 simulation for each one. So the first one we show is the RC curve. So this one is we use sample size 150, true number of covariate 15, and the total number of covariate P 200. That's one of the data set we have is having this sample size and number of the covariate. So that's why we choose this to show. So we can see from the RC curve, I want to emphasize here the RC curve, what we show here is to evaluate the accuracy of the variable selection, is not to evaluate the model prediction. So basically we know who is a true beta. who is a true covariate from the simulation. But we wanted to know with our variable selection approach, whether we can select those true variable on the top ranked list. So we compare different methods, and you can see clearly, the elicited frequency method to high dimensional inference method outperform than the other method. And for the stepwise regression and the stability, they're not performed very well. Yeah, sure.
To control the RC curve, you need a probability of being selected, right? So how do you compute the probability of being selected for elastic net, for instance?
So for elastic, because we use the frequency elastic. So frequency elastic for each covariate, you have a frequency through 100 times how many you are being selected. So if it's a strong predictor, it will be select 100% time. If it's not, it will be select 0% time. So there are a lot of zero as time, but for the other, they have a frequency.
The other one use the p-value.
For stability, it's the same. Stability also have that ranking because it's a frequency. So it gives for HDI, because every covariate has a p-value. So you use p-value as the ranking list. For univariate analysis, you also have a p-value. So you use that as a rank list. For stepwise regression, it's tricky because stepwise regression only selects several. So basically it's yes versus no. So some variable it is yes, some variable is just no, not selected. And for, yeah, that's what it is. So it's not very fair for stepwise regression, to be honest.
That's a piece where these linear interpolation from the one dot that you have to stepwise.
I believe this is a averaging RC curve. The average RC curve is incorporating 100 simulation run. So then that's why it's not that step. It is there is a package how to ranking it, getting a more smooth RC. Otherwise, it looks very ugly. Yeah. Thank you. It's a great question. So the AUC is area under the curve. So this is probably more directed to show comparing about different methods. So here it's very clear the three methods, frequency, elastic net, H2 HDI are outperformed than the other method, especially when the beta is high. And interesting is you can see the univariate analysis actually can find the true factors if the beta is very small. So that's some interesting things. I don't have a very clear explanation why it's like that. I also show this is a big picture to putting all the simulation scenario together. This one is more showing you under the non-scenario. So basically those are the null covariate, it shouldn't be selected, right? So this is basically a type of error. And then across different scenario, you can see the black one is a univariate analysis. Basically, it's showing the univariate analysis inflated the result because of there is a strong correlation structure between covariate. So if this is a true covariate, the univariate analysis didn't treating those correlation structure controlled. They just if you are in this correlation block, it will select those. So it has a lot of false positive for the univariate analysis. So that's the one thing I want to emphasize. We also showed F1 score as a sort of how we perform about the variable selection. And we can see the two HDI method have a higher F1 score. And we do something called, we also do FDR adjustment and bound-forming adjustment because HDI, technically it is sort of the high dimensional inference for low dimensional interpretation. So we shouldn't rely too much on p-value to interpret about the finding we get. We tried the bound-forming adjustment and FDR to see, okay, if we have too many of the covariate, how we do the selection. And we can see the F1 score, even you don't do FDR adjustment or bound-forming adjustment, those two is comparable performance with the if you don't do any adjustment, but still slightly higher. Why is also want to point it for the stability performance poorly. So I'm not recommending if in the real data analysis we use stability. Why it happened like that? I think mainly because of the FDR control. And you can see if we don't control FDR, then the raw p-value will be inflated. But after you control the FDR at 20%, it is this curve, this dash is FDR controlled, it's well controlled. But you can also see if you use Bonferroni adjustment, it's controlled too much. So this is the trade-off, like what would be the way we should control about the false positive and also the signal. Here is again, it's a real data analysis. Amazingly, I tried a different method on the same small data set. Remember, it only have 48 subjects, but 132 covariate. This is our original finding. We use the frequency ELISAnet. We found the 3 marker LDH, MCP1, and IL7. This is a refitted model. After we selected, we refit a model like this. And we tried two high-dimensional inference model. It has different name when it applied to Cox regression. And we noticed that the climb HDI is actually selecting exactly the same 3 biomarker. And for the decorrelated HDI Cox version, it is selected 1 more covariate. But the 3 markers we original identified is still there. We tried the stability, and you can see, so this is a frequency ELISAnet frequency table. So those 3 markers we selected is on the top ranked list. And in the stability, it selects a different IL7 at pre-linear differential, not at peak. Basically, it's like a different IL7 at a different time point. So this is very interesting. It's like no matter, I conclude that no matter which method you use, we nailed to the same goal. It means that the data is robust and you have difference of trying different models. They all land on the same conclusion. This is a Kaplan marker. We also do a smooth curve to show the change is not linear. Okay, a quick summary is like, basically we recommend the sparsified HDI method for the small sample size data analysis. I have a R Shining website. I wanted to show everyone what it looks like. So my student from Iowa State University, he helped me to build up this R Shining tool. So this is pretty straightforward to use. You can upload your own data. This is the simulated data, and you choose which model you use. This is outcome is linear, so we choose linear regression, and you choose what is outcome, and then you can run that. So after that, we can get the descriptive summary. It's basically the table 1 for most of the paper I publish. You have a summary about what's your data for all the covariate. And then we have a univariate analysis. It gives you the univariate analysis reporting the beta and the confidence interval p-value. It also has a multivariable model that's incorporating what we talked today. It has HDI desparsified, decorrelated, ELISAnet, stability, and stable regression. You can see it's run actually pretty fast. Just several seconds, it gives you the result. It also gives you a forest plot. So those are something most important in my clinical research is that a lot of times for variable selection, the stringent things is the clinician or we don't want to conclude we need a threshold. A lot of times, statisticians will make a decision, is it true or not? We give a test, but the clinician, they don't really care about you set a threshold here or there. They just want to know a relative ranking. And this forest plot is basically giving everyone a full picture. So for the publication, the same. They want to publish this other than publish, I identify 2 variable, it's significant. They want to give everyone a full picture about what's the data and the relative result of that. So that's one of the motivation why we developed this. Also, it can use for logistic regression and survival as well.
curiosity, how did the inference happen? Because my understanding of all these methods is a lot of them require you to do a wild bootstrap or multiplied bootstrap. So do you just compute the asymptotic distribution or?
Yeah, so I didn't prepare the slides of that, but this specified, I think I think it's mainly they are all based on a score test. So especially for decorrelated, there are three different versions. It has a word test version, it has likelihood ratio version, and the score test version. It's basically there is it's hard to describe right now without any reference in my hand, but mainly it is first you get the Lasso-Galley coefficient, and then you try to develop something based on the score test, and then go back to modify the beta inference.
But then to get your confidence interval, you don't need to run a multiplier bootstrap.
You don't need, yeah.
So you're using a closed form solution
Right.
to
derive that.
asymptotic distribution.
Yeah, it is.
I see.
Yeah. Okay, so the next one I collaborate with Qian at UW, and so he helped me for this tree-based method comparison. So basically, why we are doing this is because we wanted to actually one of the grant I read about the HDI, the reviewer come and see, oh, why not compare with XGBoost, because everyone see XGBoost is the best, right? So I wanted to know better about from my special setting about this small sample size, highly correlated data, and also I care more about variable selection other than the model prediction, whether XGBoost did better. So we conduct this analysis. So the quick summary is we look at random forest, it bootstrap each and get the tree and summarizing this thing together. And there are different ways how to evaluate the variable importance. And we care most because we do the variable selection. So we try to get something can help us to select the variable. And so basically we use the permutation test to get the null distribution for each variable and get the ranking of observed importance. There are other way how to basically get a ranking of that minimal depth is another way how to do that variable selection. There are one more version, conditional inference tree, that is using different than the regular decision tree. At each node, it selects a covariate with a strong association. So that's a different way. They also have a permutation test to help for select the variable. For the XGBoost, that's the main thing we want to compare with. So XGBoost, again, is very popular, and it is based on gradient boosting, and it is, get the decision tree first, get residual, and use residual to refine the tree next, and then eventually helping for the to get a better prediction. So XGBoost original version do not have the variable selection way, so Qi implemented that permutation test on that to help us to let everything comparable. More recent generation talking is BART did better than XGBoost. Some people talk about that. So we incorporating BART based on the Bayesian additive regression tree, which has good statistical property, but runs very slow because of the MCMC. Also, a newer generation for BART is called DART, use Dirichlet other than flight prior, it uses the Dirichlet prior. So also it has some statistical property that it's not based on the permutation to select the variable. It has some property to select the variable based on this rule. So to be honest, Qi knows this better why it's like that. So those are the summary table showing all the method we sort of comparing, and because we wanted to investigating about the variable selection more, so we implemented the permutation test for the method. If they don't have that, that can help us to understand how to do the variable selection. So what we do is again compare different methods through simulation and apply that on a CAR-T study. So again, the simulation setting is the same as what we did before. Basically, we use the same simulation data, but apply different tree-based method on that. Then we can compare with our regression-based method. But this two paper is done by two different students. So I haven't merged everything together. You will see I do a little bit like Photoshop things to try to compare them back to back. May not be very precise. Here though, I use a different scenario because this is another car study. It has larger sample size, but smaller covariate. So this is no longer high dimensional setting. But again, for that, we still wanted to know which variable is more important. So we choose one setting to show the RC curve. So this is for RC curve is based on different beta. So it's a weak signal or a strong signal. So you can see here, like when the effect size is small, the random forest, those two green method and the conditional random forest, those method is looks perform better than the other method. But when the effect size is higher, then quickly BART and DART, the purple and the red method outperform than the other method quickly. Yeah, for the AUC, it's very clear too. It's not only the BART and DART outperform the other method, even there is almost no variation. So the result is turned out too good to be true. Yeah, but that's what the simulation showed. And for if the signal is not that strong, you can see the random forest actually did pretty well. So here I know it's not an apple to apple comparison. I just try to align this together to see the comparison before and after. So we probably can only see like this, exactly the same setting, but compare about the regression-based method. And we can see for those methods, high dimensional inference and a lot of other methods, they did pretty well for the AUC, the variable selection is very accurate. I don't know whether it is very clear comparing here, but somehow it's casual, right? For example, this is X-ray boost. At least it's pretty clear showing X-ray boost is not the best. At least in this setting, it's not better than regression-based method. Another one is I-FAN score. This is a tree-based score, so it's clear like the BART and the DART outperform than the other method, right? Even X-ray boost is not that strong. Good for I-FAN score. Again, for regression one, and you can see the regression-based method, HDR, 2HDR and ELISAnet have a higher I-FAN score than most random forest result and X-ray boost. It's similar as BART and DART. FDR again FDR. So the FDR surprisingly, the BART, DART, and the X-ray boost method, they control FDR pretty well. I think the BART and X-ray boost I can understand because I have my permutation p-value there. But the DART, it is just have a theoretical threshold and it never changed. It's just a 0.5. So I'm still trying to understand better why that works pretty well to control the FDR. The regression-based FDR, when we do, again, having the buffering adjustment, have the buffering adjustment and FDR control, it is controlled pretty well at the DDR level. And you can see when the sample size is large, maybe not here. It's the regression-based method control FDR at the DDR level. It's in another plot, sorry. The running time is another thing very important to evaluate different methods. So we can see the purple one is BART, so it takes most of the time to run. I think it's well known because we have both permutation and MCMC. And for the regression-based, the scale is different, you know, so the regression-based method outruns very fast. So for the tree-based method, for the comparison, I would recommend to use BART or DART. the application is always very interesting. The car study here, it is an application having 164 subjects, and we have two sets of the covariate. One has 17, another has 22. The endpoint is continuous, and the original paper we published used the disparsified high dimensional inference, and we identified we identified the first three subjects, the platelet, IL-6, and the TGF beta. We applied a different method, stepwise regression and the ELISA frequency. You can see for stepwise regression, it meets the IL-6 and the TGF beta. It selects one more IL-8. And for the ELISA frequency, the interesting thing is after the refit, those four variables are not sort of significant. And then it is hard to have a conclusion why it's like that. So, when you're adjusting those four together in the one model, then the IL-6 and the TGF beta lose the significance. But that's one of the caveats for the ELISA-net or lasso method, because you need to refit. We presenting about this as what I just showed. The forest plot, those are most important because you basically know the relevance and the ranking of each covariate. So again, we applied the tree-based method on that, and the result is quite different. So the random forest method, we choose the platelet and the TGF beta, two overlapping with our finding. And for the XGBoost, it is selected platelet, IL-8. This is overlap with stepwise regressions identified, but it didn't. I also identified one more marker, but not others. For the BART, it is selected more. Again, they not significant for IL-6, but they choose TGF beta. And for the DART, it select two different covariates. So from this, I'm learning. My conclusion would be, for the real data analysis, my suggestion is you should run all different models if you can, and then you should not see, okay, because of one method, statistically we think it did better than the other method, then we choose this method. We could presenting the table like what we do here is we use different methods and there are discrepancy which we showed. So the final conclusion we have is suggesting learned from this tree-based simulation is BART and the DART method outperform the others. But BART is very slow. So and also because of the BART have some issues about the install. I feel like it takes forever to install the BART on my computer. And so somehow I think that's maybe one of the barriers other people use it. So I recommend to use DART for the data analysis. Okay, I think I only have 8 minutes. I just quickly show something I developed. So I have one of my research interests is to develop statistical software that can help other people to use. May not all the software is my own algorithm, but it's something I think is very important and useful when we're doing our day-to-day life about the data analysis. So two package I developed, one is called 12 size, it's sample size calculation. It's 12 years old and has a lot of download now. The chip test is genetic data analysis tool using chip sequencing. Now it can apply to other type of the data too. It is archived for 8 years, has relative high download. So recently I developing the Arshani tool. I just show you one, the HDI Shiny. So it's published on my website. And another is a sample, is also a sample size calculation. So those are the students helping me, and Chloe is actually also a UW grad. And Macy is a UW undergrad. The sample size calculation, very important. But something interesting to know is like the software is now free, and the software do not have the Mac version. I have a Windows computer in my office just for sample size calculation purpose. So I think it's not that convenient. That's about the motivation why we developed this sample size calculation tool. So here is my sample size calculation. Anyone do sample size calculation in your research or work? No, okay. I do it almost every day. So basically it is, different design and you input a parameter. You can choose continuous binary survival endpoint. You can choose 1-arm or 2-arm design. You can choose different tests. And you can choose one-sided or two-sided test. And significant level differently, and you get a table. And not only we get a table, I also get a paragraph of writing. This is basically saved my life as I just copy paste this and put in my grant or someone help. I also have the R code. So for us, like it's more useful if I just copy paste the code and that you can replicate that, right? I also have something called stopping rule. That's for phase 1 study. I also use that almost at least once a week. I write this paragraph about the stopping rule for a protocol. And I also have a phase 2 study. It has Siemens 2-stage design. And the phase 2/3 study, it has a conditional power, just something I frequently use. Yeah, I myself find this is very helpful. Okay. Okay, so this is high dimensional inference. We have already saw that. I think that's a useful tool too. And I have several tools for different genetic data analysis data too. I'm particularly interested in chip sequencing data now, new generation of the pattern run. Less interested in RNA related data, but I do that too. Yeah, reference. Thanks for my collaborator and their funding support.
So your biomarkers are meant to predict if a subject will not have effective CAR T or relapse? I forgot from the very beginning of the talk, like the
endpoint. What's the endpoint?
So you're trying to predict, the biomarker is like clinically you should measure these things because they'll be important to predict the future endpoint of a person.
Right, yeah.
But the biomarker itself does not tell you how to do that prediction. It's just like this variable is important.
Depends on what's the method used, right? So for example, one paper we have having identified 3 biomarker, and then we use that 3 biomarker to separate that to be high risk and low risk. So from that meaning, we just want them to know whether this patient is high risk or low risk. So we pre-specify this patient using the biomarker to define its high risk or low risk. So this is one way. Another way is pretty traditional statistical way. We build a model, right? We have beta 1 times x1 plus beta 2 times x2. We get a score. And you use this score to see if you have a high score, you are high risk. If you are low score, you're low risk. But you not necessarily need to set a threshold. So that's actually clinically frequently used. One of the things called HCTCI score. It's been published for 20 years by a statistician in my group. This one is kind of like the Bible in the transplant world. Everyone need to first calculate this HCTCI score and then decide this patient is high risk or low risk. If high risk, they go to transplant quickly. If low risk, the patient can wait a little bit longer. Yeah, so different way to use it. But from my point of view, I want that most of the CAR clinician, they wanted to know, is this biomarker important or not? Because that's related with the CAR therapy has different generation. So for example, the current generation of the CAR may be already, for example, one study I involved, it has a protocol name like 02, 03, 04, 05, 07, 08. It's already the 08 generation. every generation, the CAR T therapy itself has a modification. So it's not the original one. Why we want to know the biomarker? Because those biomarker can help us to guide us generate the next generation of the therapy. So from that point of view, which one is more important, is more relevant in this setting? Yeah.
Any other questions? No,
Yeah, I have a question. Any student available in the summer for a summer intern? No? Okay. Yeah.
So we thank you very much.
Thank you.
Yeah, you can send us an email to the graduate program coordinator.
Yeah, to be honest, I do that every summer
You do that.
and not all.